connecting to the ones that develop new varieties and understand how we do that, why we do that in the first place, and how it can affect our lives and a bit of the history of plant breeding itself. So, as Steve has said, there's 3 other people here. So as we go along, I'm going to have everybody chime in, and the other PhD students here, they'll be chiming in throughout the presentation. And also, if you want to put your questions in chat, we'll also have the other PhD students go ahead and monitor the chat with Steve and see if we can answer some questions, unless Steve wants to pause and answer a big question there. Consider it a bit of a plant breeding tech support that we'll have. So if we're going to go through the who's, what's, where's, when's, and why's of plant breeding, why don't we start with why even do it in the first place? Well, so since the beginning of agriculture, there's been a great desire from people that began actually planting and replanting certain plants to select ones that they deem are higher quality, support their communities better, and bring about a set of desirable traits that we want to preserve, increase, maintain, and continue in this process of the cyclical agriculture to support our food systems, support our communities, so on. So some of the important traits that we look for, one of the big ones you'll hear is we're looking to increase yield. The overall total amount of barley you would get from a crop per year with as minimal inputs as possible. Included in that are also important end user traits. So as Steve mentioned, for cassava and Seren's interests, improving nutritional quality, can we increase the amount of carotenoids in a starchy root vegetable, improve the taste of a squash, the color of an apple, something that's all appealing and interesting to the end user. And this extends not just to kind of fresh market traits, something that we see in the grocery store, but for processability, we're looking for something like a potato that's easy to harvest, perhaps a certain shape of potato that's easy to ship in a factory and is a lot more efficient when it's more uniform, something along those lines. And something I know that all gardeners deal with every year and are very aware of is we're looking for pest and disease resistance, myself, as well as, everybody that breeds a crop needs and is looking for sources of genetic pest and disease resistance that we can maintain in our crops to make sure that they don't get succumbed to a whole range of organisms that love the same crops that we do. And then we're selecting for local environments. We're looking to see if we can select varieties that fit a certain community, region, nation in the most optimal way to get the best end product. And then plant breeding, our baseline foundation is diversity. And there's so much natural diversity for every single crop that part of our work also is different ways to maintain, preserve, and distribute diversity. So we have a broad base of genetics to work with and a lot of desirable traits to select from. And then, above all, also for breeding novelty crops, having people engage in plant breeding, understanding that anybody can do it, that it is enjoyable, and it is something that can bring people together. So we can go to the next slide here. And just understanding that plant breeding has been interwoven into human civilization since the absolute beginning of agriculture. It's already affected our lives in such a fundamental way. And as you can see a lot of really common grocery store crops here. Well, these are all products of plant breeding across thousands of years. You can see stemming from the same plant, this wild mustard plant, Brassica oleracea, you get this whole host of different crops, including kohlrabi, broccoli, kale, Brussels sprouts, cabbage, cauliflower, all just from selection of different parts of the plant, accentuating and selecting and maintaining those desirable traits. For example, selecting on specific traits of the leaves that we're interested in ends up in the end product of kale that we see in the grocery store. And this has been something that has been around since the very, very beginning, just doing simple selection by eye, saying, this is something I want. I'm going to replant this again the next year. And these are the products that you would see from this. So let's go to the next slide here. And part of this presentation is that we are going to take you through kind of the full landscape of plant breeding methods that we would use in modern day plant breeding to support the development of new varieties. And given plant breeding's been around so long, there are so many different ways to go about it, all with different best practices, best places, ones that suit certain situations better than others. Some of the most notable ones that we're going to be talking about here today, you can see up in the top left corner here, we have all the plant breeding methods that are mainly driven by just what we say phenotypic selection, a selection by the outside appearance of the plant. And that could be something along the lines of crossing two parents, looking at the progeny and saying, I'm going to pick amongst those, or something like mass selection here, where you would take a really large set of diverse plants, hundreds or thousands, and go through, inspect and judge and debate on the positive and benefits of all of the traits that you would see in a diverse population, and select among those and continue them, plant them again, and continue from there. So this is the most fundamental and common method of plant breeding, are just doing controlled crosses and controlled selections. This is how it started, and this is still the most common method that's been used in modern day plant breeding. And in the last decades, since I would say 60s and 70s, we started getting more DNA-informed methods. So we're looking at two here, marker-assisted selection and genomic selection. And with our advancements in genomic technology to do more sequencing, to develop genetic tests for certain traits, the same you would if you were doing any kind of genetic disease in humans, looking for a certain variation in someone's genetic code, we can use those techniques to, say, screen seedlings for disease resistance. Some would have disease-resistant genes, some wouldn't, and through genetic tests, we can determine which ones have, which ones don't have a resistance gene and advance those. And genomic selection is also a newer method where now that we can do whole genome sequencing, we can use information from the entire genome to put into statistical models and begin predicting based on previous training data which of our seedlings might have better performance in the next generation. So these DNA-formed methods really just take a look at the DNA of the seedling as it is, and we use prior information to try and predict what's happening next. And what's great is it saves a lot of time and saves us, creates an environment where we can select more with a lot more information. Some other plant breeding techniques here, we have gene editing and genetic modification, which we'll also talk and get more in depth about today, where there's a range of methods to go about this. But essentially, the idea is we would take either foreign or the actual host DNA, and use different molecular mechanisms to edit the DNA to select, either insert or delete or edit in a certain way the DNA of a crop that would either give it a new trait, a new set of disease resistance, pest resistance, or turn off something that would kind of give it susceptibility to a certain disease, so on and so forth. So we're going to talk a lot more about that today. And the last I'm going to talk about is one that's less common, from about, say, 1920s on. There's also this technique called mutation breeding, where we would take, say, either a certain mutagenic chemical or a application of a certain wavelength of radiation that would induce a certain higher level of mutations and give us a higher rate of variation in the population of plants, where we would have new traits, new level of diversity in that population, where we can now select from those plants, determine which ones have agronomic properties that were interesting to us, and either use them to study and learn more about a specific trait, or release that as, after much testing, release that as a variety. So all of these methods all leverage exchange and evaluation of DNA and the effects of a DNA on how a plant would actually behave in the field, in the garden. And this gives us all different abilities to select and to choose ones that benefit us for food, for ornamentals, anything of the sort. So we can continue here. And we're going to talk a little bit about the timelines of breeding, how long it takes, what's the process, how somebody would go about selecting these plants, making pollinations, crossing, creating new populations. But it's really important to note that the actual process begins at what we call pre-breeding. And that's something my program does. Many programs also at Cornell is we do kind of the background work to supply breeders with the information, the knowledge, and the varieties that set them up to create and develop new varieties that have higher levels of yields, color, taste, nutrition. And that all starts with looking at the diversity that we already have. So if we're going to look at our germplasm, which is say a full collection of as many diverse accessions, or what we'd say unique, say in this case watermelon, as many unique watermelon as we could find, and plant out in a field, view their agronomic properties, view what traits they have that are most interesting, and select among those. That itself is part of this pre-breeding process that is so important to know what material we have, what traits we have, and with the advent of also DNA technology, we can understand how all of these are related, where they're coming from. Are they native to Latin America, to Europe, North America, and get an idea of either the domestication story and what traits they might hold. And we do that with different genetic analyses, where we would understand where in the genome, if we were looking for pest resistance or disease resistance, we have all these different methods to say, okay, we have done all these genetic tests and genetic analysis, and we have determined on chromosome 1 in this specific place, we have disease resistance. And that would lead next to developing a DNA test for breeders to use and to accelerate the process of bringing that resistance from the natural gene pool to their new varieties. And then also, one more point here is we're also making crosses with wild relatives, including our program. We're interested in closely related wild relatives of apples that have great properties that don't exist in the regular domesticated gene pool that are used to breed new apple varieties. So because it takes a long time and it's not quite the focus of an applied breeder that would make varieties for the grocery store, we would then make those crosses and supply that germplasm for them. And finally, plant breeders, growers, and consumers, that pipeline is so important that, plant breeders that have specialized in a certain crop have also really connected themselves to the community and the end user, where if you have a crop that's going in for processing apples for applesauce, potatoes for chipping, they understand the process start to finish, so they know exactly what consumers want. We know what processors want. We can cater to that in a way that is beneficial for everybody. We can go to the next slide here. All right, so the plant breeding process. So once we've gone through the pre-breeding process, which often happens in parallel to this conventional plant breeding process I'll talk about here, you can see 3 different timelines where within the first year, a plant breeder would make a cross between 2 different parents, say a potato with great color, a potato with great agronomic properties, heat tolerance, something like that. And they want to combine those traits and get a progeny or a variety that has both those important qualities. You can see for potato, for wheat, and for apple, there's a really big difference in how long we can go from that first cross to making selections and making choices out in the field, testing them in a more real world situation of how they'd be grown by a consumer or a grower, and then the ultimate release of either the seed or getting actual fruit in the grocery store. So if we're looking at potato here, the first timeline from the first cross, understanding that potatoes are outcrossers, which means that they can't cross with themselves, and they are what we say they have a high level of genetic diversity. So once you make that first cross and you plant out all the potatoes that come from that cross-pollination and the progeny from that cross, you have a really wide selection of a lot of diversity to choose from. So by the 3rd to the 6th year after that first cross, you're already able to make selections from that first round, perform field trials that are a lot more close to the actual end growing situation, and then eventually after say 6-7 years, begin to release that variety and make it available to consumers. And this is all done with just conventional crossing. For a let's say wheat or barley or oats, which they characteristically can self-pollinate, which makes them a lot less genetically diverse throughout those selections, throughout that process, because when you're self-pollinating and you're crossing to the same plant, you're using the same genetic code and the same genetic diversity. So you end up not getting quite as much diversity off of your first cross. So you would have to plant out more, make more crosses, and then from there begin crossing that individual plant to itself and begin actually making selections on agronomic traits and getting something that's more stable and uniform every year that you plant it. And then by year 3 through 6, your selections are pretty much been finished. You know which of those potential varieties out of those selections have the best chance of being widely adopted by the end consumer and used for either large commercial growing or local production, for, in Karl's case, for beer production for barley. Then we can start field trials and begin releasing that seed for companies to sell and make available to everybody. And this is very different if you're looking at a crop like a long cycle woody perennial like apples, which takes quite a lot longer to breed. And that's for a few different reasons. One, it's like potato where it can only cross to a different kind of apple, which means there's a lot of genetic diversity, and you make your first cross, you get a lot of different kinds of apples from those seedlings. And the other thing is that we have a 4 to 6 year juvenile cycle. So from that first cross, the seedling you're growing, it's about 5 years until you see flowers and fruits to make those judgments. So off the first year of crossing, we can then take those seedlings and select among those. After 3 to 6 years, we can see their fruit, we can see their traits out in the fields, and determine which of those will continue to the next round. So you would take those, replicate them, put them in 2 different environments, see how they perform. And again, that's year 6 to 15 years in at this point. And then by year 15 to 20 from those initial selections from just that 1 cross, 1 or 2 crosses, you can begin commercial trials, get this in front of growers, have the growers do tastings, see if that's something that their users would want, their end consumers, people who are buying in the grocery stores, seeing if that's something that they're willing to grow. It's easy to grow. It is something that is agronomically robust. And then that could be distributed, those clones then to grower who can then begin growing them. By the 5th year again, we could start seeing fruit, and we're 25, 30 years in. So it's quite a big difference across different kinds of crops. And we can show that the biology of the crop is really important. The selection process is really important, and the end product of what kind of traits we're selecting on are all something that are top of mind for plant breeders that are running very strict organized programs. We can go next here, and also feel free to chime in, anybody else who would like to add anything here. But the main point here is we're doing population improvement. We're making a cross. We're looking at the progeny of those crosses, seeing which traits from the parents that they've inherited, determining which combinations were best and produce something that people want to use, people want to eat, and gets people excited about the variety itself and solves the problem that the breeder is ultimately looking to solve. And it goes in this kind of parallel cycle where that first year cross isn't so linear. The first year cross, they're doing another round the next year, and it's this cycle where we're constantly doing evaluations every year and pushing the genetics towards a direction that is best for solving the problem and best for getting something to the consumer that people want. And all of these, this process has been really enhanced and augmented by advancements in 2 different areas. So when we say phenotyping, we're looking at the kind of exterior traits of the plant. And phenotyping has taken a really large leap, I would say, in the past decade or 2, where because just evaluating many plants by eye is very laborious and sometimes biased, and we want a really accurate measurement of whether a plant is disease resistant, whether it's tall, whether it's producing a lot of yield. So some new advancements in phenotyping include being able to use images and image analysis to predict plant traits. So we could fly a drone over a field. That's what Karl's doing. Karl can fly a drone over a field and get a quantitative determination of whether a certain barley variety or wheat variety in one block is performing better than another. And that's something that's really exciting and taking off.
I'll add something real quick here.
Yeah, go ahead.
Yeah, so with phenotyping, yeah, this is an image I took of our our largest trial that we have. And so those are the combines below, but in addition to barley, where I use the imaging for, you can do that with alfalfa. And I would argue it's even more efficient, because if you use like imaging on the kind of looking at how green the plot is, that kind of correlates really well with like how much alfalfa is produced in those plots to the left. And so we've been using this technology in different ways for different species to kind of do a better job of selecting which plants have, higher yield and overall quality.
All right, thanks, Carl. Yeah, so with that, I also talked about the advent of more genotyping technology. So we can sequence, we can predict the agronomic traits using DNA data, and also make DNA tests to predict for plant disease traits or agronomic traits that we're interested in. We can continue on here. And yep, I'll let Seren take over here, and we can talk about different types of varieties that you would see as a gardener, as well as what their added benefits are, how the biology differs.
Yeah, thanks. So there's a lot of different types of varieties, and the names can be sometimes confusing. So I'm going to go through some examples of what these different types of varieties are and how they're different to work with when you're breeding or gardening. So the first one, hybrids. You hear F1 hybrids. If you've ever wondered what that means on a genetic level, plants have 2 copies of the genome. Most plants, similar to people, how we have a copy of the genome from maternal parent and the paternal parent. They have 2 copies, sometimes more. And when you're crossing 2 different inbred parents, the F1 hybrids will have 1 copy from the paternal parent and 1 copy from the maternal parent. So in this case, the pink and blue represent DNA that's being inherited from the red or yellow tomatoes. So F1 hybrids are all the same because they all have 1 chromosome, 1 DNA sequence from each of their parents. You might have wondered, why can't I save seeds from F1 hybrids? And they're not the same in the next generation. When an F1 hybrid is crossed with itself or another F1, there's a process where the DNA gets shuffled, which is called recombination. And as a result, the next population has some segments of DNA that are from the red tomato, some that are from the yellow tomato. And so we call that population segregating, where there's different traits exhibited in the population. Some are larger, some are smaller. So you can save seeds from a hybrid, but you just are unlikely to have something that looks exactly like the F1 hybrid that you save the seeds from.
There's also a situation with hybrids where if you self certain species, they get something called what's called inbreeding depression. So the plant performs really poorly to the point where if you inbreed it enough, it doesn't at all. So you want to cross 2 different parents that are genetically different to complement each other, and that's something we produces the big word's heterosis, but hybrid vigor. So you end up getting a plant from F1s that do better than either of the parents by themselves. But if you're going to inbreed that, you accumulate kind of deleterious genetic defects that result in a poor performing plant. So there is like there's a very functional purpose by developing a hybrid.
Yeah, so hybrids can be advantageous in a lot of ways. It can also be useful to be able to save the seeds from your variety year after year. And so that's where inbreds can be very valuable. So if you have an inbred parent and you are self-pollinating, when recombination happens, the material is the same that gets exchanged. So when you get to your F2 variety, it looks exactly the same as the parent. So that's why we call it true breeding. If we start with an inbred that has 2 copies of the genome that are similar or identical. And so these kinds of varieties, you can save the seeds, plant them, and they'll look the same. There's also open pollinated varieties. So open pollinated, just as a word by itself, just means that the pollen is coming from bees or other insects from any other plants that are nearby, instead of being intentionally cross-pollinated by someone's hand or paintbrush. And so you can have, for example, open pollinated apples. When you have an apple tree out in the field, the bees come and pollinate the flowers. They might be bringing pollen from different types of apple trees all around. And as a result, all the progeny of the apple will be looking different. And so that's an example of the sugar bee apple variety came from a Honeycrisp apple tree that was open pollinated. So some pollen from another tree came and pollinated the Honeycrisp apple, and that gave us the sugar bee variety. You might also see open pollinated variety like on a seed packet. And that typically means that that variety is inbred enough to where if you let the seeds all cross-pollinate with each other, rather than making controlled crosses, they end up pretty much the same in the next generation. So you can save the seeds from your open pollinated variety, assuming that there wasn't a lot of contamination from other varieties coming in. So in general, you can save the seeds from open pollinated varieties because they're genetically similar. There's also heirloom and land races, which are kind of special categories of inbred or open pollinated lines. So heirloom just means it's been an open pollinated line that's been passed down for many generations and has become like a classic variety that people have saved and passed down. Similarly, land races are varieties that have been open pollinated and passed down for many generations, usually selected by local farmers. And so there's varieties that are typically adapted to the local environment and often are associated with local culture and cuisine. So these are examples of maize land races from Mexico. And land races in plant breeding can offer sources of genetic diversity, and they're often important to preserve these traditionally cultivated varieties as more and more people are switching to like modern hybrid varieties. These land races offer a lot of diversity. Some examples of reading about these terms on seed packets. So here's an example of an open pollinated variety of corn. So this means that the seeds in the packet are very similar genetically to where you can let the plants cross-pollinate each other naturally and save the seeds, and they'll be fairly similar in the next generation. Here's an example of an F1 hybrid. So this is an example of seeds that were created when two different parents were crossed. And if you were to save these seeds and cross them again, you would get a mix of random traits in the next generation. It could be good, it could be interesting, it could be poorer than you were expecting. And that's where you see some of this hybrid vigor that Karl was talking about, and why hybrids are often higher performing for that reason. Here's an example of an heirloom tomato variety, and it has this non-GMO label on here. I just wanted to point out that there are not currently GMO varieties of tomatoes or really any vegetables that you plant in your garden. So if you see non-GMO on a seed packet, you might be wondering if other seeds are GMO that you might be buying. But in general, a home gardener would not come across GMO seeds at this point in time that they could buy without having paperwork involved. Okay, there's also vegetatively propagated plants like potato. So in this case, we have genetically diverse. So there's different copies of the genome, but there's no recombination that happens, because instead of saving the seeds from a potato, we propagate it via seed potatoes And those are just growing from the same, resprouting from the same type of plant. So the genetic makeup stays the same as you propagate it. So that's a good way to maintain a hybrid plant as a hybrid, just by propagating it vegetatively.
Yeah, Seren, I could add something here. Yeah, so the important note here is vegetatively propagated crops maintain the exact same level of the exact same genetics, but over time, as you maintain the same clone of a potato or of an apple tree, it can accumulate mutations just from either UV radiation, something like that. That could either be kind of neutral and unnoticed, or actually be beneficial. And so that's where we get sport varieties of apples, or actually you have a single branch of an apple tree that accumulates a couple mutations that will give it a brighter red color or slightly different taste of the apple. And somebody can take a cutting of that, maintain that, and it'll preserve those traits. So if you see a Fuji and a Fuji red sport, the only difference is maybe a single letter difference in the genomic sequence there that will give it red versus more brightly red colored fruit.
On the other hand, with vegetative propagated plants, this may not be breeding per se, but sometimes they can accumulate deleterious alleles or viruses occasionally as well. So there's definitely some cases with, like, keeping vegetatively propagated like flowers and whatnot, where if you keep them for a while and you start seeing patterns that are different, or tulips are an example of that. With the colorations, those are the result of viruses in accumulation in the plant.
Yep, it's good to replenish stock after a while. Yep.
Yeah, so that's a good transition to note that mutations pop up in the DNA randomly through time, and there's also, and that can create genetic diversity and phenotypic diversity that's useful for plant breeders. Sometimes we might want to create that genetic diversity intentionally, or create mutations or modify the genome intentionally. And so that's where plant biotechnology comes in. There are gene-edited crops and genetically modified crops. And Marlie's going to talk more about that in a minute. There's only a few genetically modified or gene-edited crops that are being grown in the US. Sugar beets, for example, maize, soy, papaya, potato, cotton, and camelina. So you are likely to encounter genetically modified foods in processed foods in the grocery store. You're unlikely to, at this point in time, encounter genetically modified seeds or fresh vegetables in a home garden scenario. So I'll let Marlie talk more about this technology. Gets a lot of questions.
All right. So as Seren and Ricky mentioned, the majority of what plant breeders use are these kind of traditional plant breeding mechanisms, where we're either looking at the actual plant traits to make selections, or we may be looking at genes or different areas of the genome to make selections. So plant biotechnology is relatively new compared to these traditional methods. And we can break it up into two main groups. So we have what most people think of as GMOs, or transgenic plants, and then we have gene edited. And a lot of these use similar methods to actually put in or knock out a gene into a plant. But there's one really big difference. So when we talk about GMOs, genetically modified, or transgenic plants, we're talking about taking genes from something else and putting it into a different organism. So something that some people may be familiar with is there's this green fluorescent protein that jellyfish produce. So in actually like cancer treatments, they actually have taken this gene and made cancer cells express it. So we're taking something from the jellyfish species and putting it into some sort of cancer cells. So that's the kind of idea of genetic modification in the medical field. But it's also commonly used in things like corn, where we're taking resistances from different types of like bacteria, and we're putting in so our corn plant is now resistant to some sort of insect. On the flip side of gene editing, we're basically doing what breeders could do, but much quicker rate. So we're looking at a species and we're either knocking out a gene or putting in a gene that's already there that could occur spontaneously in nature or through plant breeding, but we're just targeting a specific spot and we're speeding up that process. So instead of taking, like Ricky said, 10 plus years to come up with a new variety, this could take maybe 5 or 6 years or less, depending on how long it takes to go through the process. So 2 really well-known examples of GMOs would be the rainbow papaya and more recently, the GMO American chestnut trees. So in papaya, there is this disease called papaya ring spot virus. And unfortunately, it was introduced to Hawaii and was plaguing all the papaya trees. And it got to a point where almost all of the papaya production was being wiped out of Hawaii, which is our major papaya producer for the US. So researchers came up with the idea of taking resistance from another organism and putting into the papaya. So now many of our papayas that are grown in Hawaii are the rainbow papaya, which has that genetic resistance from another species in it that allows us to spur Hawaiian papaya production to still continue. More recently, you guys may have heard of the blight that is plaguing our American chestnut trees. This work's actually being done at ESF up in Syracuse, where they are taking resistance from other organisms. I can't remember if this case is from other species of chestnut trees.
I believe this one is wheat.
From where?
Wheat.
From wheat. Okay, there we go. We're taking it from wheat. We are putting it into our chestnut trees. And as you can see here, we have our GMO chestnut trees that look amazing and our traditional chestnut trees that are no longer with us.
So the really interesting thing for the chestnut trees is that because we just target one part of the American chestnut tree, we maintain the form and genetics of everything else. One prior strategy that they have done at CUNY-ESF is to perhaps, make a cross between American and like a Chinese chestnut variety, because the Chinese chestnut variety does have this resistance. However, when you make the cross between those two, it doesn't have the same characteristics as opposed to just doing a more precise gene modification of the American.
And as Seren said, most of the GMO products that you see today are in processed foods or in animal feeds. You don't see it very common in fresh market produce. But a lot of the GMOs right now are very highly regulated and are typically licensed by different companies. So many of our corn varieties that have different pest resistance, those gene transformations are owned by a specific company and farmers have to pay a licensing agreement with those companies to be able to grow it. So there becomes a lot of paperwork, and it's mostly really large farms that often have contract agreements with seed companies and some sort of mill or something like that are using the varieties. There are a few other ones, like there is a transgenic squash, but the likelihood of you seeing this is very little to none at all. There have been some other efforts that make GMOs more towards for consumers. So one of them is eggplants in I believe it's in Southeast Southern Asia. They put the
Bangladesh.
Bangladesh. There you go.
Yeah.
Thank you. Where they have BT pest-resistant eggplant, and they made it for consumers to reduce the amount of pesticides you need to spray, because people were getting seriously ill from not having proper protection and using really harsh pesticides, as some of these countries do not have as many regulations to keep farmers and farm workers safe. So when we get into gene editing, one of the really common ones right now in the market is reducing browning of products. So this has also been done in apples and in potatoes as well, but I would say the mushrooms is probably one of the most common ones that people know. So we have these cute little button mushrooms, and basically what they did was there's about 6 genes that control this browning, which is a production of an enzyme called polyphenol oxidase. And this is something the mushroom naturally does. And when you knock out or remove one of those genes, it greatly reduces that browning, and it really gives the product a longer shelf life. So this is different, as this is something that could occur in nature. There could be a mutation to a gene. There could be some sort of recombination that changes this enzyme's production. So we're not adding any new DNA from another species into the mushroom. This is something that's totally within this species genome that could happen. And just a few pros and cons when we come to biotechnology. One of the really great benefits is it really speeds up the breeding process, and we can make really targeted changes. So when we talk about diversity and wanting to pick 2 parents that are more diverse, so we're getting whatever traits that we want, whether it be disease resistance and say yield in the same variety, we are going to get a lot of other stuff mixing up that we don't necessarily want. So when we work with biotechnology, we can focus on a very specific gene or a couple of genes that we want to target, either knock it out, add a gene in. So we are left with kind of the ideal variety we want. We can also create new traits in this, where we're taking a trait from maybe another plant species, like with the American chestnut GMO crop plants, and putting it into another species. So these are traits that we may never have had, or would have had waited a long time for natural mutations to occur to get the trait we want. Very important, again, with speeding up very long cycle breeding cycle crops like apples and other fruit trees. And we can target regions that are really difficult to change in conventional breeding. So there's certain areas of a chromosome when they cross over and create recombination that are really hard to happen because they're so close to the center. So think about like your arms are more like to cross over together at the top of your head than like way down here. It's a lot harder. So there's this idea where there's other regions where we can't really do it naturally, or it's going to take a very long time and a lot of different crosses and a lot of different breeding cycles to get there. We can do that very quickly. One of the really big cons right now is we don't have a lot of knowledge of well-studied genes where we can make those really targeted gene edits. There's a very few select number of crops that have a lot of genetic studies on them. There's certain plants that don't really like to go through the process of having genes added to them and having to grow them in like tissue cultures. So there's a lot of issue here around this. And this is really why you only see GMOs right now in things like corn and soybeans and cotton. Will it be in the future? Who knows? But we also talked about a few other genetic mechanisms that we can also use to help speed up the breeding process as well. That may be the future forward. Another big issue is regulations. A lot of these, it's a lot of paperwork, it's a lot of money, you need to have contracts and really have a large farm to even utilize these, with the exceptions of things like the papaya and the BT eggplant. So most of the time, smaller farms and gardeners are really never going to interact with these plants. And lastly, one of the big cons is consumer acceptance. I think the scientific community, when GMOs came out, there wasn't enough interface with consumers and the general public to really understand what it meant to be a genetically modified organism. And I think now with this round of gene editing, we are doing a little bit of a better job kind of helping people understand what's happening. And I think, of course, it's always the consumer's choice of whether they want to use or consume any sort of biotechnology derived varieties. But I think there is something there with kind of the science communication and making sure that consumers understand what their options are so that they can make educated decisions from that.
Yeah, so as we've done this larger overview of a lot of the different plant breeding methods, from more phenotypic based selection, conventional crossing, we talked about DNA informed methods, having genetic tests for different traits, talked a little bit about genome editing and genetically modified organisms. We, in conclusion, kind of want to state that there is no one silver bullet, there is no one best route or any one any one particular technique or method that we're really trying to hone in on. What we're interested in and what we're trying to communicate is that the future of plant breeding, being able to feed 10 billion people by 2050, being able to make sure that our agricultural system is supported, is using kind of a stratification and a cross-section of where these techniques best fit in. And we're excited to talk with you guys and learn from you and answer some of the hot questions you have in your head. So we would love to know what you would want to know about plant breeding and kind of open things up here.
I'll take this first question here, because I think I need to clarify a little bit. So I would have to talk to people at ESF a little bit more. I have gotten a tour of it, so but I guess to clarify, the GMOs are for doing gene editing from wheat. What I was referring to earlier about Chinese chestnut varieties, those are crosses between actual different plants. So there's no GMOs of coming from like explicitly genes from like from varieties that are come from kind of the Asian continent, so to speak. The GMOs are more of kind of what we want to keep is like most of the American chestnut background, but just have a specific gene for counteracting the blight that is unfortunately devastated most of the chestnut.
Yeah, this is also something that's not on the consumer market. So
yeah, this is still in the testing.
If you're worried about either getting a GMO American chestnut or not, you would know because you would be interacting with USDA, APHIS, what's the other one? FDA, a whole different set of government organizations that you'd be filling out paperwork. You'd be, you would know because you would be, you would have to go through a pretty rigorous process to even get that. So if you're able to just go to a landscaping nursery and purchase American chestnut tree, you're not going to come across the GMO variety.
So the next question here is, do breeders concern themselves with plant desirability for pollinators? I'll leave that off to people that have that request the bee services for their pollination of their crops. But yeah, Ricky, do you know anything about this? For apples, at least.
Yeah, I would say. So I'll start with the specific of apples in this case. The desirability of pollinators, so that would be things like color or certain compounds in the nectar that attract pollinators. And when you're breeding a long cycle crop like apple, you're really limited to the amount of traits that you can focus on at one time. If you focus too many all at one time, then you're going to get kind of an undesirable amount of each. But if you really focus in on, say, fruit quality traits and disease resistance, which are the priority for a lot of breeding programs, then you're going to have more desirable levels of each. When it comes to pollinator desirability, that's not so much a focus, at least for apples and other tree fruit crops, because those genes are so well conserved and so, I would say, redundant in the genome that we never really have a problem seeing like a drastic decline. And it's even self-selecting in a way where if the pollinators do have some aversion to a certain apple variety, then we would likely see a low crop yield on it. We would see a low amount of apples on the actual tree because the bees don't show up. We don't get what we want. So that's something that kind of selects in the background anyways. So if we just don't see a lot of trees on the apples, we know that there's low pollinator desirability and low crop yields, and that just gets selected out anyways. But we do try to promote pollination. So growers, breeders, will try to plant, say, like clover in the rows and try to attract pollinators because they are the hardest workers in the orchard.
I feel free for anyone to chime in, but I can answer this next question. How do plant breeders balance between the production benefits to commercial growers with the importance and flavor and nutrition for the end eater? So this has been a very important question, especially in the last decade or so. I'm gonna go with tomatoes since this is a classic example of this. So you know, there's many tomatoes that have been, you know, bred for, you know, grown in California, so you can have them all year round. And so in that sense, you have access to tomatoes all year round. However, when you're shipping them across the country, you want to breed them so they have a good shelf life, you know, don't bruise, so to speak. And so unfortunately, the cost for that is if you're going to breed for that, you may sacrifice something like nutritional quality or flavor. With tomatoes in the last few years, that focus is starting to change, where we're working on the fruit quality. But of course, the compromise is they may not last as long. And so this is kind of one reason it's important to breed for local environments, because if we have tomatoes that are bred locally here, we can get more of those better, nutritious, and like more fresh tomatoes to the local supermarket. Now, that may mean that you're not going to get those the whole time all year, but in the case of New York, we don't have that climate all the time. There are currently some growers that are trying to do this in greenhouses, and so environment controlled greenhouses with tomatoes, so to speak. Is there any other crops that people think of that there's a lot, but
I would say, yeah, the larger companies I think are mainly focused on agronomic traits like yield, harvestability, storability, and it's mainly smaller seed companies and more localized breeders who are focusing on traits like end user traits like taste and nutrition. Although hopefully that will change more as, I don't know, I hope more people will focus on end user traits in the future.
One of the largest kind of interesting aspects of my research that it's been really fulfilling is that I've been focusing on barley, and to an extent that comes to the wheat community, where we have focused on yield, and that's still important. However, we in our program we've learned that the quality is just as important, if maybe not more. So if we don't select for traits that result in the plants germinating early, the quality is ruined, or just the general kind of quality for malting for beer. It has to have the certain amount of enzymes, the certain amount of sugars. And if it doesn't meet that malting quality standard, instead of, getting $10, you're going to get $5. And that's like, you're not going to make or you're not going to be profitable on that. So you have to make sure that you care about the quality. And, more people are concerned about where their foods come from, and rightfully so. And so that's something that's going to be an ever increasing priority.
Awesome. How about we, I think Seren's got an answer in the chat for the next one, but let's just get her thoughts on it out loud here. So are other countries, this is from Scott, are other countries introducing them with local control, them referring to GMOs and gene edited crops? So Seren, if you want to add some insight here.
Yeah, so the regulations in the US are somewhat strict from the perspective of a breeder trying to get new variety out, in that an average breeder, someone at a university, would be unlikely to be able to release a GMO variety that is kind of more exclusively towards big companies that have the resources for going through all the regulations. Other countries, besides the US, are typically even more restrictive, to where some don't allow them at all. The US is considered more relaxed on its policy because it allows gene edited crops. So like the mushroom that has a mutation that could have occurred naturally, but it was made intentionally. It allows that with less strict regulations. So it's considered one of the more relaxed countries on GMO policy. And I see Daniel Hickey asked, is CRISPR technology used for genetic editing? Yes, so yeah, CRISPR is what's used for genetic editing. And is complete genetic sequencing done on selected crops pre and post modification to determine the extent of alteration? Yes, generally it is, although in gene edited crops and research papers that aren't like released as varieties, the complete sequencing might not have been done, but usually for a release variety, it will have been. But in general, off-target sites will be, like possible off-target sites will be sequenced for research purposes.
So there's another question there. Are there any problems with self-pollinating a plant that is in nature open pollinated? So they gave an example of a potted lemon tree. I do not know the genetics of lemon that well, but this depends. Ricky, do you know?
Yeah, well, so most tree fruit crops, pears, apples, a lot of them like to outcross. And a lot of tree crops outcross, and they're very genetically diverse that way. But actually, a lot of citrus like to self-pollinate, like they just naturally self-pollinate. Yeah, your potted lemon is doing excellent. It's doing its job. Yeah, and there's even cases for certain citrus also where you can get fruit without pollination. It's I forget the specific term. I think it's like parthenocarpy or something. So citrus is a bit strange in that way, but yeah.
Citrus is another fascinating example of like crossing two different citruses, and you get something like a grapefruit.
Yes, yeah, pomelo is a cross between two completely different citrus. So yeah. Okay, so we got Daniel Hickey, his questions, Seren. Okay, we're gonna go on to Keith. With all different sweet corn types, why can't or haven't they bred traditional corn varieties that maintain their sugars longer before turning starchy? Who wants to jump in on here?
I kind of answered that one there. We actually do have those varieties out there. The old normal genotypes is what were grown for years. But we have the sugar enhanced and then the super sweets. And the super sweet, they just don't convert at all really to starch. So we have those varieties out there.
I think we're at the end of the chat questions. If anyone would like to
Yeah.
unmute and ask a question, feel free.
Oh, someone asked about black barley. Oh, this is exciting. Yeah, so
Uh-oh. See
so
you in an hour.
yeah. So and with barley, with the organic naked barley, so this is kind of a barley where the hull falls off in a similar manner to wheat, and so that's primarily used for food purposes. And so with barleys, there are different colors, so it's not just your yellow, white kind of. There's blue ones, there's purple ones which have anthocyanins, and then there's black ones which is like a melanin, so it's a different pigment entirely. So yeah, I've made some crosses a few years ago, back in 2020 with some barleys that are considered black, are have that dark color, and so we're in our lab. There's going to be some of those coming out. There's another kind of grain called Emmers. They also have this darker color, and we have a graduate student this year that's working on those as well. So yes, we do work on those.
I think the next one follows up with Steve's answer. Is the super sweet failure to convert starch what gives the strange texture?
And yeah, that would be it. They're just very high in sugar, very slow conversion, very different texture when you're chewing it. And some consumers do not like super sweets at all because of that. And they should, most of it probably want either the traditional varieties or the sugar enhanced varieties. And with the high sugar content that's in the seed itself, it makes it harder to grow because of all the sugar in there. You plant it in a cool soil, and disease organisms will just feast on that sugar, and as compared to other varieties. So it's a little bit of a problem in the field as well.
Yeah, there's another kind of example where you can have certain varieties of alfalfa that have like low lignin content, and so that's great for cows to digest that. But at the same time, with the lack of lignin, that's kind of a defense mechanism, and so that results in those varieties of alfalfa being more susceptible.
Yeah, I think that's a point to emphasize here while we're getting more questions, is that plant breeding is always a dance or a balance between trade-offs and compromises between different traits. So like Karl mentioned, if you have reduced lignin in your alfalfa, you have better digestibility for cows, but you also lose pest resistance. It happens all the time, where if you're looking to have bigger potatoes, you lose above ground foliage. It's there's a limited set of resources within the plant. And then the other point also is that there's limited resources within a plant breeding program. So however many traits they can focus on at one time, or however many crops they can focus on at one time, it is I would say, an optimization problem of using your funding, whatever, say, royalties you're getting in, to put in the best and highest value places. So that's why a lot of prioritization is on yield, and disease resistance is that's what everybody wants, and that's what the market wants. So it naturally, that's where the dice will fall. So go ahead.
Next question here is, once you develop the plants, is there any quality control on seeds? Are all seeds of a particular variety of equal quality from one seed company to another? So I'm going to give first example. We're going to talk about barley that I'm working on, and then I'll briefly mention a little bit about kind of seed packets a little bit. So with barley and wheat in New York State in our small grains breeding program, there's, once you select a variety, one challenge is you got to increase the seed. So for us, we call that breeder seed, and so we're really meticulous to make sure that it's kind of true to type. There's no contamination of another seed of another variety, so we select that. Once we get enough seed, there's a program that's part of New York State called Foundation, and they do the increasing, and that is their primary job, is to make sure that the variety and the seed that we give them is true to type and it's really persistent. So with kind of row crops, this is kind of a whole process in itself. is to make sure that you have very pure seed for these purposes, or your case of corn hybrids. That one's a little easier to manage with the contamination because you're always crossing the same two. When it comes to your seed packets, companies, Johnny's, Burpees, so to speak, Igo or Organic, they have set germination tests that they do before they put them in those packets to make sure that they're high, because you can seeds that aren't germinating well, that's not good for the company. Marlie, I'm not sure if there's anything more you want to add about that.
So for vegetable seeds, they're like when you get into like bigger like volumes of seeds you're selling, there's more regulations. So there's some different like seed certifiers that you can go through. But the big things are like you can only have a certain percentage of like weed seeds in there. Usually like vegetable seeds is not an issue. But like Karl said, the big thing is germination. So for most vegetables, common ones, I think it has to be like 85 to 90% germ in that packet. When you get into more specialty crops, so I work on a lot of squashes and gourds from like Southeast Asia. The regulations just aren't quite there. So if you buy something like specialty, like from a very small like breeding program from like Etsy or something, things like germination quality are not going to be as good just because the species as a whole doesn't have great germination. But yeah, germination is really the big thing for vegetables.
Okay, I can see the next one here. I've seen potatoes available that have a lower glycemic index than the typical potato. There's a way to know how that's bred. Are there different ways to achieve that same type of goal? So let's break this down a little bit. Is there a way to know how that's bred? So there are resources available online. If you look up a low glycemic index potato, you can find some details of how it's bred. From what I know about how they bred that specifically, the starch and sugar traits for a potato are really well known. So I believe they had actually pretty good markers, genetic tests to understand before even planting out or before even harvesting the potato or understanding what the potato looks like. They can screen seedlings and give them that genetic test and say which ones would have either higher starch, different kinds of starch, or lower sugar. I'm not sure exactly the specific trait, but I do know that they use marker assisted selection for that one. And then there are different ways to achieve that same type of goal. This can be done with conventional breeding, and they do different kinds of tests in the lab to see how much starch or sugar a potato would have after harvesting. So yeah, either through conventional breeding, this can be done with biotechnology, and genome editing, as well as marker assisted selection. There's many different roads to get there, and sometimes the best one, it depends on the situation. Here, they found that marker assisted selection was the most optimal route, and they were able to accomplish their goal. Anybody else can jump in on that. I'm not a potato person myself.
Yeah, I'm not as well either, but that could be a good example for genetic editing too, because with potatoes, if you have to do, you can't, it's hard to inbreed those. And they're also this case of we were talking about like diploid and stuff, so there's a certain amount of chromosomes. Potatoes are what you call autotetraploid, and so that makes it more difficult to cross because, in humans, we have 2 copies of a chromosome, and in potatoes, there's 4. And not only that, they like cross with each other. And so you get this kind of thing called a dosage effect, and it makes it much more complicated to cross them. So that makes it more difficult. And so gene editing could be a way to help with this.
It's a bit of a genetic explosion. You get one cross and you get so much diversity off of one cross that it's hard to maintain that. It's hard to plant it the next year and expect the same thing. So if they use genetic markers, then they can have some more control over what they're going to expect. Because oftentimes, genetic mechanisms are have evolved to be randomized processes for a reason. Genetic diversity is the foundation of all our biological systems. So a job of a plant breeder is to kind of beat the casino in certain ways.
Yeah, all right, so I think we're gonna go to the potato one real quick, and then we'll go to the GMO question, because kind of similar topic. So yes, they have come out with diploid potatoes, and so that's an active area of research to make this easier. The jury's still out on how well they yield yet. I'm not sure yet. I don't know if Walter is convinced yet, but he will be soon. Walter DeYoung's a potato breeder. And yeah, he's
the barrier, the arbiter.
Yeah, exactly. And yeah, it's probably one of our most successful breeding programs at Cornell. One of his potatoes, I think, the number 3 in the country. And he uses, I think, less space than we do in our small grains.
Yeah, there's Lamoka and Winona, I believe, are I think like 1 in 3 of the chipping potatoes.
So this next question I think is quite important. So I gotta move up a little bit. I think from a consumer standpoint, a lot of people are concerned that GMOs may contribute to things like food allergies. In your opinions, do you think that's a legitimate concern? And what kind of research, if any, is being done in that area? So I'm going to separate this into 2 things. So there's GMOs for the pesticide, for herbicide tolerance resistance, put that in the one category for a minute, or GMOs, concerns about GMOs regulating directly for food allergies. So this is why there's a lot of regulations, just to make sure something like this, although the chances are low of happening, could occur. I'll give kind of like a negative example. They have done GMOs, for kind of like I think it was soybeans, and they tried to introduce a gene from kind of what is a Brazilian groundnut, and they found that the gene corresponding to this groundnut into soybeans ended up having an allergenic effect. So this did happen, and so they stopped that program entirely. So this is something that is on the front mind of when these things are done, that they get rigorous consumer testing. Now, the downside is if you have a lot of it, it's very expensive. And unfortunately, only larger companies can usually absorb that cost, with some exceptions. I think another issue with the GMOs is that there's traits associated with how GMOs are used, with herbicide tolerance. That's going to bring up glyphosate, for example, dicamba and things like that. And so, that's definitely an area of research that plant scientists do as well. But that's also why there are a lot of regulations in place to make sure that these things don't or should not happen. But yeah, these are good questions to ask.
I just want to add to that the process of plant genetic modification, it can be used for kind of any gene. So it's not necessarily the process that is a concern, but each instance of plant genetic modification needs to be evaluated on its own. If you introduce an allergen into a plant, then yes, it will be allergenic. So it's really more about what gene is being used and how it's evaluated and tested, more so than the technology itself.
Folks, we're getting close to wrapping things up. The questions are good, but we're going to have to. I think maybe the last one is the person from asking about Peru and their tubers. Did somebody want to take a crack at that? And I think maybe we'll do one more, and that would be it.
Sure. Peru is Peru and Chile and South America is actually the genetic center of origin of potatoes. And so what that means, so that's the location where potatoes were eventually domesticated. Now, the original potato's wild relatives were actually poisonous, and that's why you don't eat potatoes that are in the sunlight. They get that green color, and that's kind of a toxin, so to speak. So there's some people that I think were a little hungry that that resulted from that. But yeah, so there is a research center in Peru, in Chile, that has these, that maintains such a genetic diversity of these. So yes, it is not surprising that there's a lot of different diversity of those varieties. And so I'd have to ask more specifically about what tubers you're talking about, because we, you know, we're just talking about different starch content, different colors, a lot of potatoes that have like kind of a purple color. And I know Walter DeYoung's program right now has some varieties or test potatoes with that kind of purple analcyanine color. They make potato chips to test how well they do, and so that's kind of how I know. Yeah, so there's definitely them out there, but yeah, there's a whole diversity of potatoes that have different colors and sizes, characteristics.
Yeah, I can kind of speak a little bit on diversity of crops in the US and kind of, I guess, market projections, because that's a lot of kind of the work I'm doing with bringing crops from Southeast Asia and Africa into the US. So I would say the big thing is right now that like our lab's doing and a lot of other plant breeding labs are doing is how can we connect plant breeders to different consumers in different communities. So right now, there's not a high enough demand for certain crops from like the general public that is coming to seed companies and researchers, that's allowing for there to be a bigger diversity of food in like your big supermarkets. You're starting to see that more in places like Wegmans, but it's still pretty minimal compared to what it could be. There is, according to like this projected census for kind of the population of people who are from different, I guess, different ancestral or more recent backgrounds from different areas to be coming to the US. So I am expecting that there's going to be a higher demand in the future for more diverse crops to be in larger supermarkets. But I think that's one thing that kind of consumers can kind of build demand for, wanting to see more diverse foods in the marketplace and making them more accessible to consumers as a whole.
Okay, I'm going to call it a night. I can't thank you enough, and what I would really appreciate is for those, before we lose everyone, I want everyone who is willing to show their faces to show their appreciation, like I'm doing right now, for Richard, Seren, I'm sorry, Marlie and Karl. There we go. And of course, Steve Reiner.
I had an easy job. I learned so much.
Yeah.
Thank you so much.
But we really appreciate it. This was a very good talk, and I'm sure it was a lot of lessons learned for us. And we hope to maybe have a different conversation again another time. But thank you again, and we hope you have a good season, certainly a good year. We know you're working hard on your studies and your research, so that must keep you awake at night if you're still in school. So anyway, thank you all, and have a good night.
Thank you so much.
Right. Thank you, David. Thank you, everybody.
Very
Great
good.
questions. Thank you.
Appreciate it.
Thank you. Bye.